So welcome to Stephen's thesis defense. We are hosting Stephen's defense at an unforgettable and unprecedented time. Thanks for joining us today. I ask, oh, my thing is not working. I cannot move my slides. Let me do this.
It sometimes sticks.
Okay, I see that. So, okay, it's working. I asked lab members to submit one word best describing Stephen and received 34 submissions. Kind, thoughtful, and nice won the competition. So Dan, our single cell expert, helped me with a simple word clustering analysis and found a few major clusters and some small ones. The one cluster says Stephen is smart and wise. Smart, yes, of course. Wise? We will see if that's true. So somebody said Stephen is wise, right? You be the judge here. Stepping on the thin ice on Lake Michigan, drinking with Shohini and others. So I like to add 4 more words. In my view, Stephen is happy, collaborative, collegial, and friendly. I'm surprised that no one used my words. Thank you. So I can tell you with 100% certainty that Stephen is a great mentor and recruiter. During the past 5 years, he mentored 3 graduate students, 5 undergraduate students, and 1 high schooler. He helped them to become productive members of the scientific community. So Stephen is a cat owner. We are a cat lab with a total of 9 cats. So 2 of them belong to Stephen and Miriam. Feng and I have a lot of fish. Why they are not showing up here, Wenwei? They have probably been scared away by our lab cats. So Stephen is a student I'm proud of. Our lab was featured in the Washington People News. Stephen and I had a great time showing our wet lab work, although he's a dry person. Thanks to Bob Boston and our own Matt for pictures. Stephen is an outstanding researcher and writer. He's the main writer for his 3 first author papers, and he wrote major sections for his 2 co-first author papers. More importantly, he has been the editor-in-chief for our Dean Lab News and Fun page for 3+ years, and he semi-retired last year and has been serving as a consultant for Li Jun and Dan. He will officially retire after May this year. So Stephen is a mentor and athlete, loved by us. He beats Kwan 6-2 in both recruitment and in ping pong tournaments, which you can see here. Stephen is graduating at a difficult, unprecedented time, as I mentioned earlier. We took the precaution of wearing self-made masks during our last in-person gathering about 2 weeks ago to say goodbye to Stephen. Bye-bye, Stephen, and we will miss you here. Now I want to turn the screen to Stephen. Stephen?
All right. All right. I'm going to ask that everyone stop their video for this section so people can focus on the slides. All right. Well, Good afternoon, everybody, and thank you, Lee, for that wonderful introduction. I'm so lucky to have this opportunity to share with everyone all the stuff that I've been working on for the past several years. And I have to admit that I never envisioned conducting this defense from my own living room, but here we are. And so I'm thankful for each of you who are listening at home. Okay, let's get started. All right, so my title is a bit of a mouthful, and I want to break it down into smaller pieces. And it starts with multi-omics integration, which simply means that we bring together different data types to try to answer questions from multiple perspectives. Omics here is a shorthand way to refer to the various DNA and RNA sequencing technologies that we use. Gene fusion discovery is the focus of two of my projects, where we use large patient cohorts to detect a type of genetic change called a gene fusion. Somatic mutation haplotyping is another project where we improve the resolution of mutation studies by determining which copy of the chromosome a mutation occurred on. And finally, cancer. This has been my motivation for getting up each day to come to work. We all know someone who has been affected by cancer, and our goal in all of this is to better understand the genetic changes that lead to cancer and how to improve precision medicine to improve patient care. Right, so we've got a broad audience today, which I love, and I want to go over some of the important background concepts. And sort of the main core concept is that cancer is a genetic disease that is subject to evolutionary pressure. And when we think about this, we need to consider several different types of variation or mutations that may lead to cancer. Germline variation is what makes each human unique. It's what we've inherited from our parents. But some inherited variation may cause a predisposition to develop cancer later on in life. Somatic mutations accumulate randomly over time, either due to bad luck or environmental exposures. And sometimes these random mutations can disrupt normal cell function, leading to cancer. We can distinguish between somatic mutations and germline variation by comparing tumor sample DNA to normal DNA and seeing what's unique to the tumor. So by disrupting the equilibrium of normal cell function, mutations may give some cells a competitive advantage, allowing for uncontrolled cell growth and cancer. And so once a cell has that mutation and that gives it a competitive advantage, that mutation is replicated again and again as the cell divides, and so it's present throughout the tumor. And that's how we pick up on what sort of mutation has occurred, is we're able to sequence the tumor and see that it's different from the normal. All right, so what kinds of questions can we start to ask about tumors? So one early project I worked on combined two different data types, DNA methylation and DNA mutations, to predict whether a tumor would have a condition known as microsatellite instability. And we benefited from having multiple data types because the biological pathways that control microsatellite instability can be disrupted by both methylation and mutation. So combining them together helped to tell a more complete story. Another instance of using complementary data types helped us confirm that something bad had happened to our data. And I still get nervous when I talk about this. As a grad student, it was very unsettling to discover a large sample swap had occurred that affected dozens of samples being analyzed by labs at other institutions. But we were reassured when both the copy number profiling and TP53 mutation status agreed that these samples that were labeled as normal were actually tumor. So with those basic concepts covered, I want to use a specific example that motivates the outline of my talk today. This image is a representation of a tumor over time. The different shades of purple and blue indicate tumor clones, which you can think of as similar to a branching evolution tree. At the time of primary diagnosis, this patient had already had several major genetic changes happen in their tumor. There was a translocation event that brought parts of 2 different chromosomes together, and this led to a gene fusion and also affected gene expression. There was a deletion of an entire chromosome, which basically ripped out 1 chapter of the cell's instruction booklet. And within this context, 2 mutations occurred on a gene that is often mutated in cancer called NRAS. And I'll talk later on how we were able to use an emerging technology to suggest that these mutations actually occurred independently. Then after initial treatment, this tumor was nearly wiped out, but some cells remained, which seeded a relapse after a period of remission. And so to be able to craft a narrative for an individual patient like this, we must rely on previous studies that included thousands of patients across many cancer types so that we know where to start looking when we see individual patients. All right, and so one of those large cancer study data sets that we analyze is called the Cancer Genome Atlas, or TCGA, which included over 9,000 patients from 33 cancer types. And along with Ching Song and Wen Wei, we set out to characterize the gene fusion landscape from this whole data set, which involved an incredible amount of computational pipeline development and data analysis. So fusions result from some sort of genetic aberration, like a translocation or a deletion, that bring together 2 genes to form a new hybrid gene. And this event may give a selective advantage to a cell because creating this hybrid could, for example, cause growth signaling to be turned on all the time. So we study this using RNA-seq data, which measures the intermediate step between DNA and protein by looking for evidence that connects the 2 genes together. So for each cancer type, we ran several detection tools, filtered results so that we could be more confident, and then analyzed the results to find meaningful biological outcomes. One important observation was that oncogenes, which are genes that tend to become hyperactive or take on a new function in cancer, were overexpressed when that gene was involved in a fusion. And the reverse was true for tumor suppressors, which tend to become less active or not functional in cancer. We looked in public databases to determine which of the patients in this cohort had a druggable fusion, meaning that there was some previous evidence to say that this fusion could be targeted by a specific drug therapy. And we found that 6% of patients had 1 or more druggable events. So doing a large-scale study like this sort of reveals the landscape of events and motivated us to look in more detail at another cancer type, which was not included in the TCGA, and that is multiple myeloma. Multiple myeloma is the 2nd most common type of blood cancer, and it's caused by a proliferation of plasma cells in the bone marrow. Plasma cells are collected using a bone marrow biopsy and using CD138 positive sorting to select for plasma cells. Plasma cells are responsible for creating antibodies that help your immune system recognize foreign invaders. With that responsibility, the IGH gene region is highly active. And when translocations occur that bring that region into contact with oncogenes, those oncogenes get overexpressed, leading to cancer. And so given the prevalence of translocation events in multiple myeloma, we thought it would make sense to characterize the gene fusion landscape of this cancer type. So with data from the Multiple Myeloma Research Foundation, or MMRF, we analyzed gene fusions from a cohort of 742 patients. We combined RNA, DNA, and clinical data to analyze fusions, gene expression, copy number changes, and structural variation. For our fusion detection pipeline, we combined 5 different tools and required that fusions be called by at least 2 tools in order for us to count it as being a real event. And we applied additional filters to increase our confidence in the result. We further used DNA to support fusion events by looking for evidence that there really was a genomic rearrangement that brought the 2 genes together. And the most recurrent event involved the IGH gene, which previously mentioned, and the oncogene WHSC1. And this is the result of a translocation called T414 because it brings together parts of chromosome 4 and chromosome 14. All right, we again looked at the relationship between gene fusions and gene expression. For example, expression of WHSC1 on the right side of this plot was over 10 times higher in samples that had the fusion than in samples without the fusion. It was also interesting to detect 3 patients with a fusion involving NTRK1, which is in the green color, sort of in the middle. The NTRK gene family was the first fusion gene to have an FDA-approved drug therapy, regardless of cancer type. So these patients may benefit from this drug that was approved. So thinking about treatment and evolution of the tumor over time, we had the benefit of looking at some samples that were collected from multiple time points. So patient came in from at the primary diagnosis stage and then came in again later at relapse for another sample to be taken. And so we can compare information from those 2 time points. And there's a lot of information here, so I'll just focus on one patient, patient 1656. And when we look at the DNA mutations of this sample down in the bottom, we see there wasn't really much going on at the first visit, but there was a lot more going on at the later visit. But at both time points, we detected this NTRK1 gene fusion. With a lack of events happening in the first time point, this may be an indication that the NTRK targeted therapy may be good for this patient. All right, so another way to think about tumor evolution and tumor heterogeneity is by using single cell data. I'll talk a lot more about this data type later, but for now, on the topic of fusions, I just want to say that we developed a pipeline for identifying chimeric transcripts from single cell data. That pipeline is called Fuchsia for fusion detection in single cells. And so on the left, you see, so each dot is a single cell, and we were able to assign cell types to each cell. And the tumor cells are basically the same as the plasma cells there. And on the right, you'll see the cells in which we detected this fusion event, and it was only detected in plasma cells or only in the tumor cells. So that lets us know we're doing a good job in terms of specificity. But there are a lot of plasma cells that didn't have this fusion detected. So we think there are ways that we can later improve upon the detection power. Okay, so back to bulk sample analysis again. There's a previously reported phenomenon of 2 neighboring genes both being upregulated as a result of this T414 translocation. So in this case, IGH actually enhances expression of both gene WHSC1 and another gene FGFR3. And we see that in approximately 72% of samples, we have high expression of WHSC1 on the x-axis and high expression of FGFR3 on the y-axis. But there's also this group of patients down at the bottom where they have high WHSC1, but a normal level of FGFR3. And within this lower FGFR3 cohort, we noted a pattern of copy number loss of FGFR3 while maintaining normal level of WHSC1. Whereas in this other group with high FGFR3 expression, those samples tended to have normal copy number of FGFR3. And a related question concerns this 15% of patients with a pathogenic FGFR3 mutation. And we wanted to understand the relationship between those mutations and the translocation event. Did those mutations happen on the same or different haplotype as the translocation? So you can imagine a cell here with 2 copies of chromosome 14 and 2 copies of chromosome 4, like normal. And we have the IGH gene region here on chromosome 14 and the FGFR3 and WHSC1 region on chromosome 4. And then there's this translocation event that brings together the IGH region and this region on chromosome 4. So that's depicted in the second cell. After the translocation, there's this exchange of genetic material between the 2 chromosomes. And so we want to know where this somatic mutation happens that affects FGFR3, this orange star. Does it happen on this haplotype that was not affected by the translocation, or do we see it on the haplotype associated with the translocation? And we observed that the RNA VAF of FGFR3 mutations was always higher compared to the DNA VAF, and leading us to think that the mutant allele was being enhanced by the translocation, which would mean that it occurred on the same haplotype with the translocation. But this is just an observation, just an interpretation. So it would be really cool if we could observe this co-occurrence directly. And that's where this emerging technology called linked read whole genome sequencing comes in. It's a newer technology that retains some long-range information about the relationship between mutations. So with regular sequencing, we have all these short pieces of information without any way to link them back to the original molecule that they originated from. And with this, we can see how often the mutation occurred, but we don't know if it occurred on the same haplotype as another one. With linked read sequencing, there's an additional step which labels each read so we can link them back together. If 2 reads have the same label, then we know that they represent information from the same haplotype in the cell. We can think of each haplotype as the copy of a chromosome you inherited from each parent. So for example, we have two copies of each chromosome, one from each parent. And when we distinguish between those 2, we say there's like a paternal haplotype and a maternal haplotype. Here indicated by purple and green different colors. In cancer, you can imagine this gets a little complicated, but the same ancestral concept remains the same. So the way this data is generated is by isolating large pieces of DNA into bubbles, and which are these circles here. And within each bubble, there's a gel bead containing barcoded primers. Those primers attach to the DNA and get amplified to create DNA fragments with barcode tags. And those fragments are sequenced and the reads are aligned, variants are called, and those variants are phased, meaning that they are assigned to a particular haplotype. And this pipeline is thanks to a company called 10x Genomics. And this is their Chromium system, which we have access to at the McDonald Genome Institute. Now there's a very important concept that I want to emphasize here. So the added value of doing linked read sequencing is that we now have these structures called phase sets, which are genomic regions where we can distinguish between haplotypes and know which haplotype a particular variant belongs to. And within a phase set, haplotype assignments are consistent, meaning that two variants assigned to the same haplotype are actually thought to have come from the same biological haplotype. However, the phase set extends only so far, which is shown here by different colored boxes along the chromosome. So you have a dark box and a light box, dark box, light box. Those are different phase sets. And once we start to compare variants from these different phase sets, the assignments of haplotype are no longer consistent. So haplotype 1 in one phase set may represent the same biological haplotype as haplotype 2 in a different phase set. Right, I'm going to take a water break here, and this is a really important concept. So think about it for a few seconds, and then I'll be back on. Okay, so we have this new data type that we're looking at with phase sets and variants phased to haplotypes. And we have this incredibly rich resource that we have access to, where we have 14 patients with multiple myeloma, all sequenced with this linked read whole genome sequencing. And these 14 patients came into clinic across multiple disease stages. So from this pre-malignant smoldering myeloma stage to the primary diagnosis, all the way down to relapse. And so we have a total of 23 samples from these various stages. Some of the samples were sorted to select for tumor plasma cells, but most were not. And we can feed these samples into our new computational pipeline, which we're calling for now somatic haplotype. So we feed this phased BAM and this phased VCF into our new pipeline. And the pipeline creates a new data object to store phase set and variant data types, so they are ready for downstream analysis. And later modules include Summarize, which gathers information about each phase set, Somatic module, which systematically assigns mutations to haplotypes, which the standard pipeline is not always able to do. We have the Extend module, which uses shared germline variant information from multiple samples to bridge gaps between broken phase sets. And then we have the Ancestry module, which looks into public genetic databases for information that may be useful to also extend phase sets and identify ancestral origins of haplotypes. Right, so let's jump straight into phasing somatic mutations to their haplotype of origin. So each somatic mutation has a set of barcoded reads that support it. And we use information from reads associated with that set of barcodes to determine the most likely haplotype for the somatic mutation. One approach looks for a consensus haplotype assignment based on the alleles from other reads. This is what we call the linked alleles approach. And the other approach uses information given by the standard pipeline output, which assigns some reads, not all reads, but some reads of specific haplotype. And when we combine these two approaches, we find a tremendous amount of overlap between them. When both approaches make a haplotype assignment to haplotype 1 or haplotype 2 in this bottom left corner, it's almost unanimous agreement for these 9,000 mutations that were called. And then sometimes one method isn't able to make a call either because there's not enough coverage or it did not meet the phasing criteria. But the other method is able to, or it does have enough coverage to make a call. So one method can rescue another one. And so we looked we applied this to mutations called from our data set and looked specifically at mutations that are often seen in multiple myeloma. And so, for example, we have this one patient with 2 NRAS mutations here that were called by the other pipeline, but were not phased by the other pipeline. With our somatic specific approach, we were able to add this phasing information on top of what was already provided. So this pair of NRAS hotspot mutations were close enough to be covered by reads originating from the same molecule. And we know that they occurred on the same haplotype based on the haplotype assignment of the surrounding alleles. But we never observed them together on the same barcode. So it's always one mutation or the other, but never both. So this guides our interpretation that these 2 mutations actually occurred in different parts of the tumor, or as we might say, in different subclones here. So we've got this one NRAS Q61 subclone, which was we thought independent of another NRAS subclone with G13 mutation. And I specifically want to thank Ching-Song and Amila for this. They were the pioneers of this type of analysis where they were actually drawing out on a whiteboard all the different barcodes and seeing which combinations made sense. And so it's from their inspiration, I was able to do this, to write code to do this systematically for any pair of mutations. And so we can also look at structural variants using barcode information. With this long range information, we see the entire structure of the T14 translocation. So here, each green or gray dot indicates one read, and dots that fall on the same horizontal line come from the same barcode. So they originate from the same original molecule. The vertical dotted line indicates the translocation breakpoint. And so DNA from the right side of both breakpoints on chromosome 14 and chromosome 4. DNA on the right side of those breakpoints join together, and DNA from the left side of the breakpoints join together, as depicted in this cartoon diagram. That this part of chromosome 14 joins together with this part of 4, this part of 4 joins together with this part of 14. And so without linked reads, we wouldn't have this level of information. So we also see this deletion on chromosome 14 that is proximal to the breakpoint. And so without linked reads, we wouldn't be able to have this level of detail. Okay, and finally, how can we leverage germline information from multiple samples to add value to the standard pipeline output? So imagine we have 2 samples from the same person. And so the patterns of germline variation are identical and have consistent haplotype orientation since both samples came from the same person. When a phase set gets broken up in one sample, it might not be broken up in the other sample, and we can bridge those gaps using shared overlapping germline variants. So you can see here in the diagram 2 target phase sets that are broken up in one sample, and then in another sample, the reference phase set bridges the gap between them. On the left side, the haplotype assignments match up. See, H1 matches with H1, H2 matches with H2. But on the right side, H1 matches with H2, and H2 matches with H1. So we modeled this relationship using a bipartite graph structure with different edge weights according to whether the target phase set needed to be switched to be consistent or not. When we sum across the edges connecting 2 target phase sets, we know if they are in the same orientation or if they need to be switched to be consistent. In this way, the number of pairs of variants for which we know the phased haplotype relationship grows exponentially. So as a data-driven example, we saw several target phase sets bridged together by one reference here. So from We have 7 target face sets here and 1 reference that connects all of them together. And the graph structure shows some overlaps. We're not convincing enough to know if we can switch or not switch, but for T4, T6, and T7, there was enough overlapping variation with R1 that we could be more decisive. The inferred graph on top shows the relationship between T4, T6, and T7, and that T6 has a different orientation than T4 and T7. So overall, this approach allowed us to extend face set length nearly 5-fold compared to the standard pipeline output. And another related concept here is that we also share germline variation with our ancestors. So, we share half of our germline with each parent, 1/4 with each sibling. And farther and farther you go down the family tree, the less and less you share. But there is still some segments that are inherited identical from generation to generation. So the same concept of extending face sets based on germline overlap applies if we can identify individuals in a public database that share genetic information with you from a common ancestor, so-called identical by descent or IBD segments. So in one example from the 1,000 Genomes dataset, we found IBD segments spanning broken face sets and matched up the germline alleles and determined that the 2 face sets had opposite orientation here. So here the germline variants that overlapped were assigned to haplotype 1. Here in this other face set, they were assigned to haplotype 2 here and haplotype 1 there. So we know that they're in opposite orientation because of this overlap from the IBD segment. Right, so we've talked through how data integration helps us get a more complete picture of the cancer genome. But we've talked mostly about bulk sequencing, which blurs all the cells together from one sample. What's next, and what I'm quite excited about, is that we can do all these same type of analysis, but at single cell resolution. And this is the final topic I'll touch on today. I was lucky to work with Raiyang and Qingsong and many others on this expanded data set from the same 14 myeloma patients mentioned earlier. The difference here is that we focused on single cell RNA sequencing data, which allowed us to examine the coevolution of tumor and immune cells across multiple disease stages. So how can we think about single cell RNA? I like this fruit smoothie analogy. So in bulk sequencing, we put all the cells in a blender, and what we consume is a mixture of all the different signals. But with single cell, we retain information about each individual cell, which gives us an understanding of how each cell individually is behaving. And so on the bottom left here is a typical single cell analysis plot. Each dot is one cell, and dots that are plotted close together have a similar gene expression profile. And based on prior observations, we know that the expression patterns of different cell types, so we can assign clusters to be particular cell types. And what this lets us do is examine how cell populations change over time. For example, the pink cells here represent tumor plasma cells. At primary diagnosis, they are the most prevalent cell type, but later at remission, when the tumor is dormant, they are nearly wiped out, only to return again at relapse. By tracking single cells over time, we can observe more precisely which subpopulations of tumor cells survive during remission and cause relapse, which is of great clinical interest. And again, of course, we can integrate multiple data types at single cell resolution. In this sample, there were 3 populations of plasma cells shown in the teal circles here, we have cluster 1, cluster 2, and cluster 3 of plasma cells. And so we examined FGFR3 expression, chromosome 13 copy number change, and NRAS mutation status. And what we saw was that in cluster 1, none of these tumor-associated changes had occurred. In cluster 2, there was FGFR3 overexpression and some copy number 13, chromosome 13 copy number loss. But we did not observe the NRAS mutations in that cluster. However, in cluster 3, we observed all 3 of these cancer-associated events. So we're able to sort of draw a more interesting narrative on how these cells may be related. So this sort of data integration at increased resolution is one future direction that I see for myself. My vision is to create data analysis tools that help people understand their data, contextualize their findings, and then translate that into better patient care. All right, and with that, I want to do some scientific acknowledgements. To the myeloma team at WashU, it's been a pleasure to learn from you what all this data really means. And it was so special to me to be involved with the Rhino Blood Cancer Research Initiative. So thank you for including me. To my committee, I can't wait to chat later. Thanks for your help and guidance throughout this process. Thank you to DBBS and the Human and Statistical Genetics Program for bringing me to WashU and providing administrative support and fellowship along the way. Thanks to Precision Medicine Pathway. Those were really fun lectures that got me really thinking about how to connect technology with patient care. And thanks to YSP and the Teaching Center for keeping me on track with professional development toward my career goals. And finally, wow, thanks to Lee and the whole Dean Lab for your constant inspiration. Your bright personalities and intelligence made those everyday moments feel extraordinary. And it's really been a pleasure. So thank you. All right, I imagine everyone is breathing a sigh of relief and clapping now.
Yes, we're all clapping.
All right, thank you, thank you, thank you.
Now we are open for questions.
Yes, I'm happy to take questions now, and once questions are over, I'll go back to a set of personal acknowledgements. So I guess if you have a question, just start talking and identify who you are. Thank you. Thank you.
First, unmute. Wenwei, you have a question? Oh, maybe we can. Do we see questions from chat? You can post your questions through chat as well.
That's right.
You can also use the raise hand feature.
Okay, so I guess I will ask committee members if you have questions for Stephen.
And we'll have the private section later.
Yes, we do. But if you have questions, feel free to ask.
Saving our ammo for the private session.
Okay. So I guess people are starting to get used to this this type of thesis defense. So I guess it's a little bit difficult to ask questions.
Hi, Steven, I have a question.
Okay, Hoshang.
Hi, very good talk. So can you go to your slides, like the last several slides? There are and you analyze the plasma cells by single cell.
Mm-hmm.
Yeah, can you go to the last?
Sure. Am I sharing my screen? I forgot.
I'm
yet.
not seeing your screen.
There we go. Okay, can you see it?
Yeah. I think it's here. So how and we see there you did some the gene expression and also the copy number
Yes.
number analysis. And and I was wondering, how did you do the somatic mutation for each single cell? Here?
Right, so that's a good question, and it's a more detailed process than in the normal talk. But so if we know just from another data type, like from whole exome sequencing, that this G13R mutation is there, we can look, and this is a coding mutation, so it's possible that it would be covered by the RNA-seq reads from each cell. So we're able to look for the specific mutation at the RNA-seq. And I would encourage you to use Song's tool, which is amazing for doing this.
Okay, it's kind of based on the RNA-seq analysis, right?
Right, so yeah, since that's the sequencing data that we have from each cell, it's only the RNA-seq, so we're limited to looking for coding mutations. And even with this data set that's biased toward the 3' end of each gene, we're kind of lucky to be able to find this G13R.
Okay, I see. Thank you. Mm-hmm.
Do we have additional questions from people on the call? Okay, I guess it's an excellent presentation, very, very clear. And so Stephen, maybe we can move on to the next set of slides.
Yeah, I'm trying to get my, okay, here we are. I just want to make face contact with everyone and just say thank you again for bearing with us through this difficult presentation process, and I appreciate it so much. So thank you. And then committee will show up on the other call.